So today we're talking about RNA. What is RNA and how is it related to the process of transcription? To talk about transcription, we need to keep in mind the central dogma of biology. If at any point during protein synthesis, which is the next few sets of notes, if at any point during these notes you get confused, come back to the central dogma, which at this point you should know is DNA. The arrow should be right here, but it didn't get moved up. DNA, RNA, arrow, protein. Information starts in the DNA. It is then transcribed to the RNA and then translated to the protein. Information starts at DNA, goes to RNA, goes to protein. And here, this picture right here is going to help you. Information starts at the DNA. The process of making RNA is transcription. The process of making a protein is translation. Today, we're talking about transcription. This whole process right here is called protein synthesis. Today we're just talking about transcription. So let's go to the next page. So protein synthesis occurs, actually it occurs in all of the cell cycle, but let's think about this. It's going to happen a lot in the G1 phase and the G2 phase because we're getting ready during the G1 phase for synthesis, for DNA replication. We need to make those DNA polymerases. We need to make those helicases. We need to make all of those proteins and protein enzymes to help make these processes happen. And so in G2 phase, we're making enzymes to make cell division happen. So while it does happen through the whole thing, primarily it's happening a lot in the G1 and G2 phase. So let's go on. In the process of making RNA, there are a few differences or a few changes from DNA. So 3 things that you need to know that's different about RNA and DNA. Well, they're both nucleic acids. Deoxyribose is not found in RNA. So the D stands for deoxyribonucleic acid. RNA is ribonucleic acid because it's the sugar ribose in RNA. So it's still a nucleic acid, but a different sugar, ribose. DNA is double-stranded. It's that really cool double helix shape, but RNA is single-stranded. It is a single-stranded message. And DNA contains thymine, while RNA does not. This gets a little confusing when we're lining up complementary strands, but RNA does not have thymine. So if we ever see an A, the complement is U, and we'll practice this a bunch. The job of RNA, or of mRNA, is to run a message from the nucleus out to a protein. So DNA is the information on how to make proteins, but it stays safely in the nucleus. It doesn't leave. But proteins are made out in the cytoplasm, where it's dangerous for DNA to be. So what we need to do is we need to make a message. So we make messenger RNA through the process of transcription. So that messenger can leave the nucleus, go to the ribosome, and be a message for how to make proteins. The job simply of mRNA is to send messages of genetic information to the ribosomes, because your genetic information is nothing more than a set of instructions on how to make proteins. And ribosomes are where proteins are made. There are 3 different types of RNA that we talk about. We talk about mRNA, which is the main one that we're talking about. That's messenger RNA. There's also tRNA, which is transfer RNA. We'll talk about that during translation. And rRNA, ribosomal RNA. We're not going to talk too much about that, but do know that it exists. So transcription, simply the process of reading DNA to make RNA. It's right here. So DNA is transcribed into RNA. Now understand that transcription, or to transcribe information, is that it's speaking the same language. So DNA and RNA are both speaking the language of A's, T's, C's, and G's. If I were to talk and somebody were to write what I was saying, they're going to be writing it in the same language. They'd be transcribing what I say. However, RNA to proteins, this is translation. These are two different languages. So RNA is in the languages of A's, T's, C's, and G's, but proteins are in the languages of amino acids. So that information is translated by a ribosome. So keep that in mind, that DNA and RNA are transcribed. It's the same language. And it occurs in the nucleus because that's where the DNA is. So that should make a lot of sense. So if transcription takes place in the nucleus in a eukaryotic cell, you should already know that it can't possibly take place in the nucleus of a prokaryotic cell because prokaryotes don't have a nucleus. It would take place in the nucleoid region. Enzyme RNA polymerase is very similar to DNA polymerase. So DNA polymerase synthesizes or makes DNA during DNA replication. So RNA polymerase polymerizes or synthesizes RNA. It reads the DNA template strand and assembles a complementary RNA strand. It makes RNA. Transcription is very similar to replication, except that you're making RNA, and there's no thymine in RNA. There's only, there's uracil instead. So a new mRNA strand is formed in the nucleus and sent to the ribosome with instructions on how to make a protein. And this is transcription. So here, this is the DNA strand. It's being uncoiled and unzipped, and this is the RNA polymerase that's reading it, assembling a complementary strand. You can see wherever there's an A, there's a U, not a T. But where there's a T, there is an A, because there are A's in DNA. So that messenger RNA is made, and it's sent out of the nucleus and into the cytoplasm where there's a ribosome. So let's do a little bit of practice on the next page. So here, if we were to transcribe a DNA sequence of ATGAC, the RNA strand would read, make little lines, A would be U, T would be A, G would be C, A would be U. Because there is no T, G would be C, and C would be G. All right, let's go to the next page. Let's try it. All right, so here we are. Let's do little lines. T would be A, C would be G, G would be C, A would be U, because there is no T, A would be U, G, C, T becomes A, and C becomes G. So let's do one last set on the other page, on the next page. All right, here we are. Let's make little lines. T, T, A, C, G, A, T. A would be U, G would be C, C would be G, C would be G, G would be C, T would be A, T would be A would be U, C would be G, G would be C, A would be U, and T would be A. If you didn't understand that, go through and try those again and practice, because you'll need to know that. But have a great day, and I will see you in class.